Hello, everyone, and welcome to phyloseminar.org. Today, as you are no doubt aware, a little more than a year ago, there was a series of events that led to the current epidemic of Ebola hemorrhagic fever. Since that time, there have been over 26,000 cases and over 10,000 mortalities. The epidemic still rages in Sierra Leone and Guinea. As part of this, the molecular evolution of the Ebola virus has been a subject of considerable interest by researchers and the general public. Today, we're lucky to have the first talk in a series of two talks by a pair of collaborators, Danny Park and Gittes Gudis, who are key parts of a larger team really driving our understanding of the transmission routes and the evolutionary trajectory of Ebola. Today, we're going to hear from Danny Park, who will give an overview of their recent research in the field. Danny got his bachelor's and master's degrees in computer science from MIT, where he developed software tools for in silico protein design in the Keating lab. He then worked with the Sebeti lab as a software engineer at the Broad, and then as a Harvard graduate student, and then finally as a computational biologist at the Broad. Welcome, Danny, and I'm looking forward to your talk.
Thanks, Eric, and thanks for having me here. I'm new to this group and to this format, so let me click a little bit and get started here. All right. And then I will try to full screen this thing. Does that work, Eric?
Looks great.
Okay. So as Eric mentioned, I'm the first of part of two talks, and I think a lot of the oh, this is interesting. Let me try to slow it down a little bit. Maybe I'll just full screen this and just go like that. I think a lot of the interesting parts of our project will probably, especially for this audience, the molecular evolution of the virus and things like that, will actually be presented by Gittes in a couple of weeks in June. But I'm going to focus on kind of the bigger picture of the project, the collaboration, the consortium, and the types of questions that we were trying to answer with this. So my name is Danny Park, as Eric said. I'm part of the Broad Institute, also affiliated with Harvard, and the work we're going to present is part of this larger consortium at the Viral Hemorrhagic Fever Consortium. Ebola doesn't really need much of an introduction. Within a few months of the outbreak last year, it made headline news in a lot of different ways and captured a lot of global attention, especially as it started to spill out a little bit out of the continent and into some Western countries. This is probably an older graphic. The numbers at the top are a little bit up to date, but as many of you are aware, the magnitude of this particular outbreak just outstrips all of the previous ones combined, and just the geographic reach is much larger, and even just its focus, where it's located, it was different from where the Ebola virus had been seen before. And so there were a lot of questions about, is there anything different about this particular virus? Where did it come from? Is it the same one? And that kind of thing. So these are a lot of the early questions that we and others tried to address with our work. Before I get a little too much into the Ebola work, I thought I'd just introduce a little bit about why our group in particular, how we got involved. The group that I represent, Pardis Sebeti's lab, primarily based at Harvard and the Broad Institute in Boston, is a lab that's been focusing primarily on the question of, let's see here, there we go, primarily on questions of human evolution, using genomics and computational methods to search for signals of selection recent in human history. This is where Pardis Sebeti herself kind of got her start in research, and one of the things that you find is that a lot of these signals are intricately tied to infectious disease kind of selective pressures, and so we also had a lot of interest in applying these same types of methods to the other side of the equation, the infectious diseases themselves. I got my start applying a lot of computational genomics methods to the malaria genome, and looking for signals of adaptation within malaria. And a large section of our group has been dedicated to the study of Lassa virus, both the viral genomics as well as the human genomics kind of surrounding that. We got interested in this virus. Lassa virus was generally not very well known outside of the kind of smaller community that studied it. But we got interested in this virus because of signals of selection we found in the human genome back in 2007. These were signals of selection and adaptation in human populations that were specific to a part of Nigeria. And it appeared to be involved in mechanisms that had to do with viral entry, particularly for Lassa virus. This was an area that Lassa was endemic. And so this is kind of what launched a lot of our research, both on the host side as well as on the viral side, and where we really kind of got invested in the viral kind of wet lab biology, as well as the kind of genetic analysis of these types of viruses. We don't directly handle live virus in our facilities over in the U.S., but we worked with our field sites to be able to do a lot of safe field collection of the vector rodent, as well as the virus itself, and set up a whole system to be able to study the virus, to process, to sequence RNA virus safely and effectively, and to look at kind of a lot of the kind of informatic challenges of studying highly diverse RNA viruses and to be able to do the genetics well. This also just involves setting up and kind of bolstering a lot of our field sites that we were collaborating with, and the two were primarily in Nigeria and Sierra Leone. And so that's how, what we're showing here are actually a lot of these pictures are from Kenema Government Hospital in Sierra Leone. And some of the little upgrades that they had in terms of just the laboratory equipment, they were able to set up a lab there that was able to diagnose and process a lot of Lassa virus samples that were kind of going through the area. And so that's how we ended up intersecting with Ebola when it came into Sierra Leone. As you know, Ebola in West Africa last year really got its start in Guinea, but in the area of Guinea that was kind of close to all three of these countries, Sierra Leone and Liberia, where there was a lot of cross-border traffic. When this happened back in March, well, it happened earlier, but when it was first kind of detected by the scientific community in March, we immediately knew that it would potentially become a problem in Sierra Leone, and so. And also that our Kenema Government Hospital site would be on the front line, given that it's basically the referral site in that entire region for viral hemorrhagic fevers in general. And so it would, as it processed, continued to process Lassa cases, it would probably be the one site that first would encounter a potential Ebola case. And so two members of our team flew over there with additional reagents and primers to add kind of Ebola detection to the capabilities of this lab. Did a little bit of extra kind of biosafety upgrades as well as just supplying the lab with enough materials to be able to process a larger amount of different things. The Kenema Government Hospital continued to test hemorrhagic fever samples for 2 solid months from March through May, and were able to detect Lassa, virus, but didn't detect any Ebola up until the end of May, when the ministry lab there was able to detect the first kind of confirmed case of Ebola virus. This is the actual gel using PCR diagnostics in Sierra Leone. This is actually, I might kind of pause to say that this is kind of a unique moment in that historically Ebola virus outbreaks, well, they've all happened in Central Africa, they've all been pretty localized in these remote jungle villages, but the standard story with how those unfold is there'd be some mysterious disease kind of going through community, and then usually it was always a Western team that would kind of drop in to do the diagnosis, to do the contact tracing, do a lot of the outbreak management. And this really probably represents the first time I'm aware of historically where an African scientist working for the Ministry of Health of their country was able to do the detection of the initial cases. And they followed up with contact tracing teams that they had already as part of their normal Lassa work to figure out kind of upstream where this came from and what events led to this. In doing so, they, within a couple of days, were able to identify about 50, I think it's 49 suspected cases that they decided to test. 14 of those cases ended up testing positive in their lab. And so what they did is they shipped us over at the Broad and Harvard, they shipped us all of these 49 samples, the 14 that they confirmed as having Ebola, as well as the 35 from patients who were sick with fevers that did not have Ebola, or they didn't test positive as Ebola, for us to confirm and to do some genetics and that kind of thing using a sequence-based analysis. So they, after that initial batch, within the next couple of weeks, really the cases started pouring in Kenema, and we started receiving several, I think 150 samples in total, representing most of the cases in the country for the first couple of weeks. And we went ahead and processed these as quickly as possible to try to characterize both, what it was as well as what it was doing once it entered the country. And we pushed out within a little over a week all of these sequences publicly on GenBank. These ended up being very complete genomes, not just full-length assemblies, but because this was a kind of deep next-generation sequence-based data, we were able to go in, hit it with about 2000x coverage, independent libraries, and all this kind of stuff that allows us to do to kind of accurately call intra-host variants within the virus. This is something that we had experience with our Lassa work in the past, and so we just kind of applied this same approach to be able to get that kind of data as well. But I wanted to take a moment to pause a little bit before, whenever we've kind of presented this in the past, a lot of times people were pretty impressed with how quickly we were able to produce the data. And before we get a little bit too self-congratulatory about our efforts, I wanted to ask the question, why are we doing this in the first place? Because, for those of us here in the kind of, phylogenetics community, it is worth asking the question that when a major event like this happens, how does our piece of the puzzle actually kind of assist in the efforts that are going on? And so for us, a lot of our questions became motivated from a more practical manner. We were trying to ask the types of questions, what can we learn from these viral genomes that actually can inform someone in the ministry or someone on the ground, not just the scientifically interesting questions, but also the kind of more practical questions. And some of these practical questions really have to do with where the virus come from, and how is it different, and how is it transmitting. A lot of transmission-based type questions end up being a little more applicable. And so, looking at the data now, I'm going to spend a few slides presenting things that were primarily reported in our science paper last year. But I will also, towards the end of this, start to touch on some newer data that we've been processing for the past couple of months and have released publicly, but we're about to report the analyses of. Some of the other stuff I'll leave for Gittes, but if you bear with me, the next few slides are things more from the analysis we did last year. One of the things I said early on was that one of the biggest questions was, where did this virus come from? Because people did not think of Ebola virus as a West African thing. It's generally thought of as more of a Central African thing. And so the question is, how is it related and where does it come from? What I'm showing here is an unrooted phylogenetic tree, so we're just looking at genetic distance. And I don't know if you can see the mouse pointer here, but you can see a lot of genetic distance between each major outbreak of Ebola virus that's been shown. And within each outbreak, you can see a little bit of diversity. So, for example, I'm not sure if you can't see my mouse pointer, then in the 2007-2008 outbreak, you see a couple of little hairy things kind of coming off of this little branch here. This one only had one sample. These other outbreaks have a little bit of diversity. And then down on the bottom left, you see our 78 genomes or 99 genomes that we posted online, and their diversity. but the vast majority of viral diversity is actually separating the different outbreaks that have happened. Now, a naive approach or interpretation of trying to root this type of phylogeny will tend to try to root just based on the biggest distance, and then kind of conclude that perhaps the longest branch is actually an outgroup. But what we found, and this is kind of in confirmation with previous analyses by some of our co-authors, was that if you actually try to root the tree temporally, remembering that in fact, unlike looking at perhaps like kind of a mammalian evolution timescale where all of your genomes and your sampling is all contemporaneous, for these types of outbreaks, for microbial data sets, a lot of times the data spans quite a bit of evolutionary time. And so a lot of times the longest branches are in fact the longest simply because they're the most recent, not because they're the most outgroup. And so we found actually a very good correlation between root to tip distance when we did this type of rooting and the actual year of the outbreak, and we found that this explanation of the tree made the most sense in terms of interpreting the data. And in this type of rooting, what we found was that this particular outbreak of Ebola virus in West Africa was distant from the others, not because it's special in any way, but simply just because it is the latest, and you might expect as much for the next one after that and the next one after that. In particular, when you start to look within the outbreak and break the phylogeny down, within the outbreak, what I'm showing here is a tree of the initial data set that we collected. So these are 78 unique patient genomes from Sierra Leone, all kind of shown on the right. And then I'm also showing a rooting with 3 genomes from Guinea collected earlier in the spring, published by a different group. And what you find is that the Sierra Leone outbreak, first of all, is related to the Ghanaian one, so they all kind of came from the same source. And in particular, we can see how it got into the country, and we described it initially as an initial entry into the country with 2 viral lineages in a single event. And the reason that we described it that way is because the epidemiology showed that there was a special funeral that occurred that was when the virus came into the country, that happened at a certain date. That date was thought to be early May. And based on the diversity that we saw in the sampling, and the time of the most recent common ancestor estimate for all of the samples that we saw, which looked to be kind of more like in April, we figured that the ancestry kind of predated the initial entry event. Actually, since this time, we've learned a little bit more information, and it seems like that the initial funeral event was actually in April. And so the story simplifies a little bit in a way that you might expect, which is that it looks like there was probably just a single entry of a single lineage back in April, and it was really just late May that we started to detect it. And by that point, we actually see some diversity. The other important thing point that we were making from this was that it did appear to be descended from the Ghanaian outbreak, and in particular, we saw evidence of just continued human-to-human transmission. And really, actually, this kind of gets back to that initial question that we posed, which is why, what types of analyses are we pursuing, and how is that actually helpful to folks who are involved in the control effort? And one of the outcomes that personally I thought was the most helpful was, early in the Ebola epidemic of last year, there was a lot of conversation in the public health community about the risks of exposure to the animal reservoir. People eating bushmeat, people being exposed to animals, bats and things like that, and really trying to figure out how to control people's exposure to these types of potential sources of the virus. But one of the points that we tried to make a little more strongly from our findings was that this was all human-to-human transmission, that exposure to the reservoir is perhaps what starts an outbreak, but really what makes an outbreak go on, at least for Ebola, is not exposure to the reservoir, but it's human-to-human transmission. This isn't true for Lassa virus, but it seemed to be true for Ebola virus. And after those findings were published, we saw a kind of a decrease in the just kind of conversation about exposure to the animal reservoir and things like that. And I think that was actually a productive change. I think that's one of the more practical outcomes of a study like this, and one of the things that I think is more helpful.
So just to be clear, the reason why you can say that is because you see the significant tree-like structure that corresponds to things that you see on the ground as far as human events?
Yes, so the reason that I say that, and part of it is, you can see it here, if this is a tree of everything that was known at the time, all of our Sierra Leone data plus the 3 Guinean samples, you'll see that we can root I mean, it's a pretty wide HPD, but the potential time for the most recent common ancestor of the entire outbreak is definitely within 2014, which means all viral diversity can be kind of explained by events that are actually future from the epidemiology later traced the whole outbreak to events that happened in December 2013, and so there doesn't appear to be additional diversity coming from the reservoir. Part of the reason that we think that the reservoir is very diverse has to do with this graph. Has to do with the fact that it seems that each new outbreak is sampling from some sort of reservoir diversity that's out there. In particular, the fact that these branches for each outbreak have a little bit of length to them suggests that at any given moment in time there is diversity in the reservoir that we can't see. And so if there was to be another exposure, I would expect to see a much longer branch come out. That's where that's coming from.
Got it. Thanks.
Yeah. One of the things about an outbreak where you have one kind of entry of a lineage and then it expands is that the population genetics start to look very simple. You're basically, an outbreak is something that essentially starts with an NE of 1, a really tight bottleneck, and then any new diversity just kind of gets layered on top of that. And so, we showed this figure of just kind of new different, what we're showing here is just like kind of viral haplotypes on a row. Each row is a sample and just different types of SNPs coming on, and everything seems to really kind of layer cleanly on top of previous diversity. What that means is that trees are very easy to construct. There's no real ambiguity as to which event happened first. Everything really, at least in the early days of the outbreak, seems to kind of fit an infinite sites model in a way that viruses normally don't do that. But within an outbreak, in an early outbreak, it does. And so it becomes very easy to interpret, who maybe came from who and where this, those types of transmission inferences and kind of look at viral turnover. When we looked at this initial data, we kind of saw, only a couple of SNPs that had high minor allele frequency, and so we kind of named these clusters or clades or whatever, but we basically said there's a couple of different groups of viruses that kind of share a common haplotype and maybe have some other diversity on top of that. Here's this clade 1 and clade 2 and clade 3. And if you plot those over time, we just tried to show that there's just a turnover in lineages as time goes on. The virus is not static. It's constantly just kind of dropping on new mutations in a kind of neutral manner. And over time, you see less and less of the ancestral haplotypes and just the more derived ones just kind of becoming more popular over time. In particular, not only do they follow these kind of temporal patterns, but they also seem to follow different spatial patterns, and we're able to kind of talk about some of these in more detail, but you can see that the geographic distribution of some of this genetic diversity kind of changes depending on where certain alleles are more popular in certain parts of the country than others. Up here in the upper right corner of Sierra Leone is where the kind of virus first entered into the country, but then down here, this is a health clinic where a lot of the other, the initial kind of spread of Ebola in Sierra Leone kind of got started. And then over here is the Kenema Government Hospital where a lot of the stuff got treated and diagnosed. I mentioned earlier that we also sequenced down to 2000X to look at variants within the host. This is useful for a couple of different reasons. I mean, intra-host variants, we've played around with this before in terms, and other groups have too, in terms of looking at things like viral evolution and things like that. But one of the things that we found it useful for in this particular study was kind of adding a little extra genetic diversity to each sample to be able to trace transmission events. Because one of the things that we found in this view is that despite the fact that new mutations are coming in, they probably come in at a rate roughly less than, the average rate of new mutation is less than 1 per transmission. So you have people who have genetically identical looking virus all the time, even though some time passed between them. And so the consensus assembly alone doesn't give you enough resolution to kind of separate out who may be distinct from whom in a transmission network. What we found is that looking at intra-host variants helps kind of give you a little more diversity to work with to be able to trace some of these a little bit better. Most intra-host variants appear to be kind of novel de novo events within a host that is unique to a host and never gets seen anywhere else. But every once in a while we saw a higher frequency one that got spread through a population that we believe for various reasons represented not independent de novo events, but actually transmission of variation from host to host, which is useful because then it helps you kind of build a story about how certain patients may have been infected by other patients. Now, early in the outbreak, there was a decent amount of epidemiology for each patient. For each case that came in, the ministry outreach workers were able to figure out, oh, this person had contact with that person, and they went over here, and that kind of thing. Pretty quickly, within a few weeks, as the case numbers just kind of got overwhelming, the data starts to drop off on that type of traditional epidemiology. And the case that we tried to make with this was that genetics can fill in the gap, because if you have the genetic data that ties together, two potential patients as part of a transmission chain, and then you have maybe some partial epidemiological data that says, well, maybe this person started here, and we know that they drove here, and then went off to here, and then we can link them together with the genetic data, with the markers that appear to imply some sort of shared transmission. That actually helps to fill out the story. And so a lot of this was really trying to make the case that the genetics can kind of, in combination with the epidemiology, bolster the story of where transmission may have been happening. This particular case is pretty important because it describes how the virus probably got out of this remote village into a major center and then potentially off to the rest of the country. I'll talk a little bit about some of the evolutionary analysis that we did with our initial data set. I'm going to refrain from talking too much about our new data because I want to let Gittes talk about that in a few weeks. But I'll talk a bit about it here, in particular because this is a portion of our last study that sometimes was a little bit misunderstood. But what we were looking at is the accumulation of new mutation throughout just a couple of weeks of time within the outbreak. And what we're showing here on the left is the fact that the estimated substitution rate appeared to be a bit higher than if you looked at a longer term rate that included all of the outbreaks from the past 30 years. If this is if the left curve is an estimate of the substitution rate over a very long period of time, over many decades in the reservoir, we started to think that perhaps this higher rate was an effect of relaxed or incomplete purifying selection. The fact that the purifying selection that is normally at play, reducing the rate on the long term, didn't really have a chance yet to weed out all of the non-synonymous mutations that might be slightly deleterious. We kind of see further evidence for this by looking at the long-term, this is the between outbreak non-synonymous to synonymous ratio. This is just looking at the ratio of SNPs that we see in the between outbreak branches versus the within 2014 branch, which looked a lot closer to 50-50. I mean, not quite, but almost. And the fact that this ratio almost appeared to be the same as the within host, the intra-host variant ratio of non-synonymous to synonymous SNPs, which kind of seemed to suggest that the selective pressure that reduced the non-synonymous rate might actually be weaker in the short term, or that it just hasn't had the time to eliminate things that were slightly deleterious. So this was kind of a hypothesis that we just kind of made from looking at the initial 3 weeks of the data set. Obviously, at the time, there's no way to really test this until you actually add more data. But basically, it predicts the idea that as you add more and more data from a longer and longer outbreak, more spanning a longer set of time, that this rate would be expected to come down as purifying selection starts to take its effect. And I'll just say that is something that we see, and a lot of other people have seen, have done more recent Ebola sequencing work. And this final figure is kind of, in a way, it's just kind of stating the obvious. It's just looking at a cumulative total run of how many different, the solid line here is just the case count over time in Sierra Leone, going from 0 up to, at the time, about 78. That's, in the first 3 weeks, that's all we had. But in that period of time, there was also an increase in just seeing new SNPs, new derived alleles come into play, and new haplotypes for the virus, just kind of increasing in the population. And in a way, this is just it's kind of an obvious figure, but it is just to make the point that the virus, diversifies over time. This isn't anything necessarily adaptive. It's just kind of it's moving in a way that any kind of any virus would. So at the end of all of that, basically, that's kind of where we left off. We also looked very briefly at just the impact of some of the mutations that we found in potential diagnostics and some therapeutics that were based on looking at nucleotide sequences and things like that. But we found and this was kind of what we hoped would be true, is that in the coming months after we released our study, many other groups started taking our mutation data, our diversity data, and used it to update a lot of the diagnostics and the vaccines and the other types of therapeutics that they had been working on. Which is great, because this is actually an area that, it's exactly the type of way that you would hope genomic data would be used, but it's not the kind of thing that we do. So it was good to see that this type of data has been useful for a lot of the groups that are doing the more practical work. I'll just say, kind of on the side, that in addition to sequencing Ebola virus, the way that we process the samples let us look at all the other microbial stuff too. We make a point of trying to deplete human genetic content, but we're left with all the other microbial content in there. And we didn't have a particularly huge data set, but we started to play a little bit with looking at co-infections of other common fever, or not even fever-causing illnesses in the region. And you see, as you'd expect, malaria and HIV and some other types of things prevalent in the population. And in particular, we found that some of these things, like malaria, the prevalence of malaria was actually roughly the same amongst people who were confirmed to have Ebola versus the ones that were confirmed to not have Ebola. And the point that we were trying to make here was that there were a lot of times where a person who was suspected of maybe having Ebola, has a fever, checks into the hospital, would get tested for something more routine like malaria, and then they would turn out positive for malaria, and the interpretation from the doctor and the health officials might be, oh, well, now we don't have to worry that it might be Ebola. And our takeaway from here was actually those two seem to be entirely statistically independent. Detecting one does not rule out the other, and so this type of metagenomic work, there's a lot more that you can do and a lot more you can mine in there. And we've started to try to turn a little more attention to the types of patterns you can see with co-infections and comorbidity and all that type of stuff. But the data is there, and there's a lot that could be mined as time goes on. This is not work that I was involved in, but a number of other our collaborators have also collected some more clinical metadata around the patients that were involved in our study, and tried to look at things that correlated both with infection. So if you don't have the ability to do a molecular diagnostic to tell whether or not someone has Ebola, maybe you can look at some of their symptoms and see if there is. Unfortunately, the only real symptom that seemed to correlate with it was body temperature, which is, as you know, very non-specific. You could have anything with a temperature, but it was unfortunately the only thing that came out. But we did see a number of other correlates with outcome. The types of kind of metabolic and kind of laboratory tests that a doctor might have available to them as they work on a patient. This can help them triage what types of patients are more advanced or where they might want to spend their time on in a kind of a resource limited setting. Now I'm going to turn more to some of the since that time I was just talking about what we did with the first month essentially of data from the outbreak, and I want to get to the question now of why do we keep sequencing? We had a little bit of a pause in our group for a couple of months because after the outbreak kind of got a little bit out of hand, a lot of organizations came in and things got a little bit complicated. And so our ability to do genome sequencing slowed down a lot. But then we picked up again more recently, and we started sequencing a lot of more recent Ebola samples from Sierra Leone again. And then the question becomes, so what is the additional data? How is that helpful? If it's clear, perhaps, why the initial sequencing of a virus at the beginning of the outbreak is informative to a public health effort, now the question is, why is kind of continual sequencing throughout the outbreak informative? And I'm just going to show a little bit of the data set. If that tree that I showed you earlier is basically this red tree plus this kind of light pale green tree, I'm sorry about the colors, and that's basically what I showed several slides ago. What I'm showing now is additional data that we've sequenced for the next several months in this kind of darker bluish green color. And these are all kind of, the x-axis is anchored to time, so this actually kind of shows you the temporal sampling that we've kind of expanded. I've also shown here 5 genomes for Ebola virus that were sequenced by other groups. These happen to be viruses that were taken from patients, healthcare workers, who were treated in other countries, in the UK and in Switzerland and in Italy. These are usually Western healthcare workers who were either medically evacuated from Sierra Leone, but all of these people were known to have come from Sierra Leone, or their virus, they know that they were infected in Sierra Leone. And their host country treated them, they all recovered, and they also sequenced the virus and published them online. Without our additional data, what you end up is these 5 viruses end up having these long branches, and they root into our old data set. And what you can find is that based on some of the derived alleles that are specific to Sierra Leone that we described in our old study, you can determine that all 5 of these viruses came from Sierra Leone because they contain derived alleles that are specific to Sierra Leone. We saw them emerge during our last sample set, and so it's pretty definitive. But I will kind of pause to say that you didn't need to sequence those viruses to know that they came from Sierra Leone, because these happen to be healthcare workers that you know exactly where they got infected. In fact, in 4 of the cases, you flew them out of the country. But by adding our additional, more recent data, this is the argument for why we continue to sequence during an outbreak and not just at the beginning. You can fill in a lot of the missing ancestry for these particular samples, and you can get a lot more resolution for perhaps what led up to how they got infected. And so this is just kind of like an example of taking that last tree and just stripping the tree down from, removing any of the samples that have shared ancestry, but just looking at the ones that actually seem to have, for lack of a better word, exact ancestry to the genomes that left the country. And so what we're showing is just kind of the samples that we have in our data set that have a genome that looks entirely ancestral in some way to the samples that were exported from the country, and then tying those to kind of known locations for those samples to infer the historical path, the kind of the migration of the ancestor of the virus that eventually was sequenced in Europe. And so you can see that in some cases, as this virus kind of makes its march from east to west across the country, in some cases, like in the Italian doctor's case, you can see that that virus had an ancestry that took a more northern route through the country, whereas with this Scottish case, you can see that it took a more kind of southern route through the country. So part of this is just kind of this exercise of, can we use the intermediate sampling to fill in some missing information about just migration of the movement of the virus through the country. It's not actually that important. These particular 5 samples are not what I'm interested in. It's just this exercise of being able to reconstruct the movement and just using these particular tips on the tree as a example of that. I think actually some of the most interesting stuff, both for this audience as well as just in general, for the continued sampling and data set and the analysis that we've been doing recently, actually have to do with the evolution of the virus. And so I would be very excited to tell you much more about what we're seeing and studies of purifying selection and things like that. But I want to leave a lot of that to Gittes, especially since he did pretty much all of these analyses, over at Edinburgh in the lab of Andrew Rambo. So I'll just throw up a few teaser pictures here, and then I'll take it away, and I'll let Gittes kind of fill in the mystery later. But the bigger picture again of just this kind of question of, so why do we continue to sequence virus? It's to look at transmission, to look at within country movements. There's some other stuff I didn't cover about like how it moves between countries and between demographic groups within a country and that kind of stuff. And also, a lot of the interesting stuff actually has to do with viral evolution once you have a data set now that spans a longer period of time. Because the point that I will make about the viral evolution is that it is an interesting thing that Ebola, in the past, whenever it's existed in the human host, has always existed for a relatively limited amount of time. A few months, maybe a year, but that's about it. And based on the branching of the phylogenies that we showed before, each of those lineages appears to go entirely extinct. And each time an outbreak happens in humans, it seems to be a fresh importation from the reservoir. And so what that means from an evolutionary point of view is that each time an outbreak of Ebola happens in humans, it's always a virus that's kind of, I think the word I tried to use is evolutionarily naive. It's a virus that hasn't seen humans before. The thing that's a little bit unprecedented about this epidemic is that it's spread so far and so long that it's actually, we definitely, none of our analyses that Gittes is going to present shows any kind of signs of adaptation per se, but it's been around in the human host long enough that it's no longer, what I would call naive in that sense. So anyway, these are all kind of some of the reasons why the continued sequencing is useful and this continued type of analysis is useful. And the last one that I often kind of forget or to mention, but is actually critically important, is the fact that just the simple diversity data that we produce ends up being utilized pretty quickly by the downstream groups who are doing a lot of this more practical work with vaccines and therapeutics and things like that. One last thing I'm going to just mention before I close out is this question of why is it that our group should be doing the genetic sequencing? Why should the Broad Institute be sequencing the virus? Is it because we're better or faster or than other groups? And actually, the argument that I'd like to make, and this is something actually that a lot of people in our group have been working towards, is that perhaps we shouldn't be the ones to be doing this in the future when the next outbreak happens. This quote over here from Christian Happy was a quote that a reporter got from him in an interview when our science paper was published last year. And Christian Happy is the kind of PI that oversees this larger collaborative group that we work with called the African Centers for Excellence of Genomics of Infectious Diseases. And what he is referring to in this quote is the idea that we've been building for some time, this effort to increase the capacity of research institutions, academic research institutions in Africa, to do this type of disease work, sequencing, and surveillance on their own, where we come in as a collaborative partner to assist and help prepare a lot of this stuff. But really, the future is one where we expect that they would be able to kind of continually monitor their own countries for circulating infectious disease and the diversity, and to be able to do the sequencing in-house. In fact, this whole effort was really, well, it had been in play for a couple of years, but it really got officially launched right before the Ebola outbreak was first detected last spring. And it involved a lot of the key collaborators at Kenema Government Hospital in Sierra Leone, as well as our academic institutions in Senegal and in Nigeria, and all over the world. And this is just to kind of remind ourselves that this was actually the set of scientists who did the initial detection. It was these ASCID scientists who did the initial detection of the virus in Sierra Leone as well as in Nigeria, which I had mentioned before as being kind of unique, but we hope in the future will be much more normative, that it would actually be kind of the ministry or the academic institution employees that would be able to be the ones that do the detection and the casework and follow-up and all that kind of stuff using the kind of protocols that they are familiar with. As a part of an effort to bolster a lot of this in-country capacity to do this type of work, we had been planning this for a while, but it got accelerated a little bit when there was attention from the Ebola outbreak. And we were able to deploy in-country sequencers, Illumina sequencers, both in Nigeria and Senegal, with plans to deploy those as soon as well as in Sierra Leone and in other sites as well. And this is really just a part of being able to increase the ability for these institutions to understand and do the characterization and to really know what's moving through their country on an infectious disease level. As part of that, our group, our role at the Broad Institute at Harvard, we see ourselves as part of all this, being less the ones that do the sequencing and more the ones who develop good methods and really share them and distribute them properly in a way that is accessible not just to our African scientists, but also any other collaborators that we're working with and folks we picked up along the way. So for us, that means not just, on the wet lab side, not just publishing a methods paper in genome biology about how to do the sample prep, but actually bringing people on board and having more kind of intense training sessions, whether they're from Western institutions or from our African institutions, and just showing them everything we know about sample handling, RNA extraction, library construction, all that kind of stuff, and to do the actual sequencing. This is actually a visit from CDC and Institut Pasteur folks who came to our lab last fall to kind of really pick up a lot of these types of methods that we were doing. On the computational side, this isn't just about open sourcing software and saying, here's the code, go do it, but also trying to make it as easy to run as possible in a variety of different computational platforms, whether you have a single computer or whether you have a whole compute cluster at your disposal, but also trying to translate this into a more easily accessible cloud compute kind of platform. And so we partnered with DNA Nexus to be able to provide kind of a lower cost to startup type thing where someone can play around with our methods without really getting too invested in compiling code and setting up their config files and all that kind of stuff. It's pretty easy to just throw in a couple of sequences, and if you have Ebola sequences, and to give it a try. And this basically implements the methods that we have been using for our kind of basic computational genome assembly and all that type of stuff. And I'm not going to touch on this too much, but at the end, a lot of this, just to close the loop, has to do with not just processing data, but getting some basic insights of the data back into the hands of people on the ground so that they have a faster cycle time to kind of understanding what is going through their country. Because the way we see it, what happened early last year, where we are the folks in Kenema were able to quickly get out and do the contact tracing, do the diagnostics. They sent us samples, we sequenced it, we pushed it out quickly. That was great, but the slow part was, getting samples out of the country, and especially, in the following months, this whole like kind of depending on us to do the sequencing actually seemed a little bit more of a hindrance than an asset, and so that's why we're trying to push as much of it into the field as possible, so that these insights can happen faster and they get in the hands of the people who make the decisions much more quickly. And so for me, I know that this was perhaps a little bit less technical on the phylogenetic side than this audience is used to, and a lot of that's because I intentionally left that for June. But hopefully that gives kind of a nice big picture of the project and what our, I guess really what our motivations and our thinking was behind why we did what we did, and why we pursued those types of things, and also where we see ourselves headed in the future. The acknowledgement side is incomplete, and could be, I could make smaller font and I could have thrown in a whole ton of more names in here. But it's an attempt to really just kind of capture that this is an effort that's involved a lot of people for a long period of time. And by far, the people who've paid the greatest cost are the ones that we've collaborated with over at Kenema Government Hospital. Last summer was an incredibly stressful time for us because, the folks that we lost in the hospital were not just, folks who were doing the collection and the field sites and the patient care, but these are people who have been close collaborators of ours for 7 or 8 years at this point. For some of our other collaborators, they've known them much longer. And it was a big loss when a lot of our staff started becoming afflicted by the virus as well. So that's all I have in terms of content, and I guess, I don't know if Eric wants to ask for questions or anything like that.
Yeah. Well, thank you very much, Danny. That was a very interesting talk. Yeah. So if you do have questions, go ahead and use the Q&A app to ask us some things. So let's see. We do already have a question from Peter Ralph. How big is the genome?
So the genome is not that big. It's just under 19 kb. It's like 18,000, I don't know, 600 or 700.
But it is a big virus, it seems. I mean,
yeah, I guess on the virus side, it is a little bigger. It's bigger than Lassa. It's a single. It's not segmented, so it's just one segment.
And just while we're on such things, I have heard that sometimes it's one shape and sometimes it's that long filamentous shape.
Yeah, that, to be honest, is a little bit outside of my area of expertise, so I'm not able to.
Okay.
Yeah, that I don't know.
So I was really interested to hear that the sequencing that you did helped people update the vaccine. What can you say a little bit more about that? I mean, were they just looking at sites that were under purifying selection that they could then target as epitopes, or what was the idea?
Yeah, so this is not something I'm the most familiar with because there's a number of groups working on vaccines, and it's a pretty competitive space actually. And so, but what I have learned just from hearing some of their presentations is that they have made it a point to update a lot of their kind of in-lab tests to the latest kind of lab strain that's available from the circulating outbreak, as well as using our genetic data for the polymorphisms to kind of examine each of their epitopes where it may overlap or may not overlap. In particular, the most sensitive to these are going to be some of the therapeutics that actually not working at the protein level, but looking at the nucleic acid level. So some of these kind of siRNA or other type of therapeutic approaches, because a single SNP there, even if it's synonymous, might be an issue. And what was interesting last time I heard an update from those types of groups is that part of the advantage that they pitch to their platform is that they can update the sequence very easily, and they did. And so they managed to update their whatever they were targeting, their little chunk of genome, to either the latest sequence or to kind of hit one that didn't have SNPs in it or that kind of thing.
That's interesting though that I mean, yeah. So even though there is this SNP level diversity I think you said that there's no evidence of adaptation to humans, or is that?
Yeah, so the adaptation question is a tricky one. We definitely felt with our initial data set, which comprised, just less than 100 genomes, it was just a couple of weeks worth of evolutionary time, that we were never, no matter how hard we tried, we would never really be able to pull the signal out of the noise for any kind of, positive selection test. And that definitely was true, last year. And with our expanded data set, we never saw any signals of positive selection. We looked in a few different ways. I'll let Gittes talk a little bit more about that, in a month. But it's not something that we've, nothing's jumped out at us.
Interesting, because I mean, like you say, I mean, but these SNP differences, they appear to be neutral, I guess is the point. Like, even though they're changing the epitopes and so on, it doesn't appear clearly that those are like immune escape mutations. Even if you can't just sort of run a genome-wide scan and look for the NDS.
Right. So that's actually, I mean, that's the kind of question that I think, is really important. So in terms of the neutrality, one of the things that we've found with the expanded data set over a longer period of time is that there does seem to be just an increase in the strength of purifying selection to kind of weed out some non-synonymous variants. And so what that means is what ends up remaining is probably neutral, and what is absent is probably, in some way deleterious, right? It's not something we saw in the earlier data set in just the first couple of weeks where things just kind of explode unchecked, but it's something that we're starting to see now as we've kind of gone over a longer year. Gittes has a pretty good breakdown of just evidence for why we think that's true. So.
Yeah, right. I mean, I guess, right, the population expansion, that does make it less likely to have a lot of stuff.
I see a question on the Q&A about sequencing the reservoir. That's it's a challenging thing, because one of the interesting things about Ebola is that compared to a lot of other viruses, a lot of things are not very well known about the reservoir. In fact, it seems like, as far as I understand, and I'm actually kind of new to Ebola virus myself, but it's not definitively known what the reservoir species really is. It's thought to be in some sort of bat. People have seen, I think, seropositive bats, but no one's actually isolated a virus, a live virus from a bat. And so, it's hard to sequence something when you can't even find it in the first place. I think part of it is, doing an effort like this requires enough funding that someone goes and just shoots down a whole ton of bats where a very large percentage of them don't have what you're looking for. So there isn't much data from the reservoir.
But you would actually need to shoot them, and you couldn't just take their poo or something?
Yeah, I mean, I don't think the reservoir works, so I'm just making something up.
And I mean, I don't want to just be harping on this functional side of things, but I mean, have there been functional differences observed between strains?
Yeah. So again, this is not my work, because a lot of this, I learned a lot at the last kind of filovirus conference of all these groups that do both the in vitro and the in vivo work on Ebola, and a lot of them have been asking that exact question: is there anything different about this year's virus at a molecular level, at a kind of an infection level, that kind of thing? And what's interesting is that while they're able to report some sort of change, they don't know if it actually means anything. So, for example, in the same way that as a geneticist I can report all the mutations, but I am going to be pretty cautious to say whether or not they actually have any kind of functional implication. I saw a very thorough presentation by someone doing molecular work on some of the mutations that were described in our study, doing allelic replacement on a bunch of different kind of positions within a gene of interest, and finding that there were, I don't know, some sort of changes to the ability of the virus to enter a cell, with one mutation, but then it gets balanced out with another mutation, and ending with the kind of inside of, but not sure what this really means in the host. And then I've also talked to people who do infection studies on primates in BL4 labs, and have characterized kind of qualitative differences in the nature of their pathology, but not sure whether that actually translates to humans. And then even if you knew that it made a difference in humans, you don't know if that actually makes a difference in the epidemiology as a whole. So I find that each area of expertise is pretty cautious to say what it might imply downstream of it. But,
yeah. I really liked how you were described everything in terms of like what is needed to drive interventions and so on. As far as bringing things down onto the ground, as sequencing in Africa and so on, do you think that is going to lead to more trust with the authorities? Because I know that sometimes there's been misinformation about the nature of the epidemic and how you go about getting it, and is part of the hope that the African scientists will have a little bit more authority when it comes to describing what we think from a biological perspective is going on?
Potentially so within their own countries, and in the sense that some of the mistrust simply comes from the fact that unknown outsiders are coming in to kind of make a statement about the implications of some sort of work, or some recommended course of action. And part of it is that at least kind of where it matters is trust within some of the ministries of health and the people who make policy decisions about kind of how to inform control efforts and things like that. And at that level, these types of institutions will probably only be successful to the extent they influence those types of policies. And so from the beginning, that's part of it. And so these guys are people who have pretty tight relationships with their respective ministries of health. And so, we were kind of looking for places that were already established institutions, mostly academic institutions, that had that level of relationship already, and some sort of kind of good starting place where there's an institution, a potential place for students to come in and trainees. And a lot of it actually is kind of focused on this idea of building graduate programs that didn't exist in terms of training kind of research and scientific talent that stays within the country.
Very cool. Good. Well, it looks like that's it for questions. So we'll just have to say thank you very much, Danny.
Thank you.
That you doing this, and we will look forward to Gittes' talk on the 10th of June.
I think it'll be very exciting. Thanks. It's been a fun format.
All right. Thanks, yeah.